In this EOC review, we're going to learn how to do the FOIL method so we can fill out Punnett squares properly. So here's our standard. Use Mendel's laws of segregation and independent assortment to analyze patterns of inheritance. If you don't know how to use the FOIL method and when to use the FOIL method, it's going to be pretty hard to do. It's a very simple concept once you have it figured out. So we're going to explain that to you right now. So the FOIL method, when do you use it? You use it when you're crossing two traits. So here I have an organism that has two traits. He has a dominant and recessive trait for the letter B. He has a dominant and recessive trait for the letter S. So we have two different traits here, one represented by the letter B, one represented by the letter S. Now we said in meiosis, during sexual reproductions, there are four haploid cells that are made, and you're going to wind up with 4 pairs of letters. And here's how you do it. You do it by using the foiling method. So we start with the letter F, and the F stands for first. We start next with the letter O, and that stands for outside. We then go to the third letter I, which is for inside, and L, which stands for last. Now, if this sounds familiar to you, it should. It's something that is seen all the time in algebra. You're doing the same thing, except for this time, instead of using numbers, you're using letters. So let's talk about the first pair of letters. We take our first letters. My very first letter in the set of B's is the capital B. My very first set of letters in the letter S is the capital S. So I wind up with capital B, capital S. I then move to O, which stands for outside. I'm looking at my outside letters, which is my capital B and my lowercase s. These letters are on the outermost edge, all the way to the left and all the way to the right, which is my capital B, lowercase s. Then I move to I, which is my inside. I'm going to have a lowercase b and my capital S. They are the most inner letters right there in the middle. And then I move to L, which are my last set of letters, which are going to be my lowercase b and my lowercase s. Now I have my 1, 2, 3, 4 pairs of haploid cells because they're only carrying half the letters. They only have one B it's carrying and one S that it's carrying. If you looked at our other video, you may have seen this. Now you know exactly where these letters come from. If you look right here, we have a capital BS, capital B lowercase s, lowercase b capital S, lowercase b lowercase s, and we did that by using the foiling method, and that's how we got these letters right here across the top and the letters right down here across the side. We just used the FOIL method. If you have any questions, please post a comment and let us know. We'll see you guys next time.